Hello everyone and welcome to the episode 4 of Python programming for biology, medicine and genetics, a very fantastic journey. I'm Hamid Reza Bolhassani, PhD of computer engineering, researcher, chief data scientist and founder of DataBox. I'm very happy to see you again in this episode. Okay, as you remember, we came to this, we already created this data frame all info. You remember? If you don't remember, you can review the previous episodes. Also have a look at this reference that I already shared with you in the description. So we wanna in this episode, in this fantastic, interesting part. Because why I say interesting, because visualization is always the most important, the most interesting thing and most fantastic thing for me. So in this episode we want to visualize the data and give meaning to them. So in this episode let's review how to visualize the data. As you remember, in R there is a very strong tool named ggplot2. So what we can do in Python for visualization. You know, there are also very good and lots of strong tools in Python. So since we are solving the problem from the same problem in R, so we wanna check which are the equivalent tools in Python. Here we want to create a box plot. In R the code was this. So let's see how in Python we can recreate this code and how it is converted. Okay, first of all let's go to this code. I've already shared this Google Colab link with you. First of all, we should add some other libraries. Okay, so we need, in this episode we need numpy. Import numpy is a numerical library for Python as np, and then two very great tools for visualization: Matplotlib and Seaborn. Matplotlib.pyplot as plt and Seaborn as SNS. Okay, that's right. And then we go here. You remember all these we had yeah, here we have we go through many things till now, and we came here until all info. You see, this is the data set, the data frame that we created in the previous episode. So in this episode we want to visualize this, the samples by the counts. Okay, in the x-axis we have sample and in the y we have count. And again, remember that the samples was this. We had 12 samples from basal cells and luminal cells. In each category 2 samples, so totally 12. Okay, so let's go here and see what we can do. So here for creating box plots we should use a Seaborn library, as I show to you in the beginning. I already imported that library. So we will before that we wanna define the size of a figure. So here plt as for plot figure and fixed size equal to 10, for example, and 6. Okay, then run it. Yeah, it will define the figure size for it. Already defines the figure size for us. Then we will use Seaborn library to draw a box plot. SNS.boxplot. First of all, what is the data? The data is all info. Then x-axis is sample, as you see here. Sample, yes, we wanna have sample, and in the y we wanna have count. Sample and y count. Okay, then have a run. Yeah, we see this and it's working. Yeah, and since you see, because some genes have a very high count, so it's better that we get a logarithm base 2 of this data. Yeah, like this. Yes, logarithm 2 of this, and we will see that the visualization would be more clear. Okay, so let's define here. I should define a new column named log2 of count. Okay, so all info, the new column log2 underline count equal to np.log2. Yeah, and which data? Yes, all info, and here count. Yes, yeah, I think it's right. Let's check. And it gives us a warning that we will solve it later. So let's see that what is the result. We expect to have a new column. Yes, log2. You see that some values are infinitive here and not defined, because also because we had some counts equal to 0, that we know that log is not defined for 0. So here we can have +1. Yes, log2 count +1, and here we will add a 1 here. Yes, and we will see here the problem is solved. Yes, so again let's we will copy this box plot and check the new. Yeah, we see that. Oh, sorry, here again I write count, but here we want to say log2. Let's copy this. Yeah, and check. That's right. Okay, we are seeing this as expected here, like this. You see some samples of genes. We had 12 sample of genes, and these are the logarithm 2 of counts. Yeah, logarithm 2 of counts we see here +1. In the later videos we also it's not the finished version. We want to go step by step, so but we should take these steps for more getting more clear and have some experience on the data. Okay, so but we want also, you see here we have the sample and also this x-axis and y-axis, but we also can have the, for example, plt.x label if we wanna write another label for it, but here we know it is the sample. Also plt.y label, again log, for example, log2 count +1. We can write it like here, like this. I think it's better. And also we can have a title for this title, log2 of count plus 1 by samples, and the type of diagram boxplot of log2. Yeah, you will see, yes, this is the title, this is the y-axis, and this is x-axis, but it's not finished yet. We want to make it more clear. It's just the first step, because this is the first episode that we cover the visualization. The visualization is very important in the world of data science, and here that we are covering bioinformatics and genetics and computational biology. Visualization, I think, one of the most important thing, because it gives meaning to the data and we can extract some meaningful information. Till now we came to here to visualize the data, okay, but in the next episode we will color them to make them more clear. As you see here, it is more clear to check which of the samples have the more count of genes, and also some other analysis that we will see in the next videos. I hope that it would be useful and informative for you, and in case of any questions, feel free to contact me. If you had any comment or idea or suggestions, it would be a pleasure to hear from you. And if it was interesting for you, kindly introduce this episode to your friends and colleagues who are interested. Hope to see you in the next videos. Bye.